Good afternoon, everyone. Let's get started. It's my particular honor today to introduce Natalia Umaña for her celebration seminar for her promotion to Associate Professor of EEB with tenure. Natalia received her Bachelor of Science and Master of Science degrees from the Universidad de los Andes in Bogota, Colombia. And she received her PhD in Biological Sciences from the University of Maryland. After that, she received a postdoctoral fellowship from Yale University, where she spent 2 years before coming to the University of Michigan in 2019. Since in her time here, she has received the very prestigious Jasper Loftus Hills Young Investigator Award from the American Society of Naturalists. And in 2023, she was elected as an early career fellow of the Ecological Society of America. Natalia has published over 80 peer-reviewed papers. 12 of them are in ecology or ecology letters. She's also received over 30 competitive grants or fellowships to fund her research. And Natalia has made several novel contributions to the field of ecology. She's best known for showing that variation in plant traits within a species can be as large and as important for community assembly as is the variation in traits across all species. So that reputation has garnered her 22 invited seminars and presentations in the last 5 years alone. So we're thankful you could fit us into your schedule. And Natalia has also done a tremendous amount of work helping early career scientists, especially in the area of tropical research. So I could go on, but I think I've given you a flavor of why we're so fortunate to have Natalia as a colleague. Please join me in welcoming her and celebrating her with this seminar.
So thanks so much, George, for such a nice introduction. Can everyone hear me okay? So, I don't know how to change that. Now? Is there a way? Now? It's better? Okay. Thanks so much for the introduction, George. Today I'll be sharing some of my recent research on functional ecology and tropical forest. The title of my talk is Why So Many Slow Species? Rethinking the Trade-offs in Tropical Forest. So like Ed said, I work in these type of systems. This is a photograph from a tropical forest in the Amazon, perhaps one of the most diverse, if not the most diverse forest in the world. But beyond this high taxonomic diversity that probably all of us are well aware of, I want to emphasize another angle of the diversity of this forest. And for that, I'm using this other slide. So beyond the taxonomic diversity, the high taxonomic diversity that we observe in this tropical forest, these systems also exhibit a great morphological and functional variation. So here, each of these different squares represent a different species. And what I'm showing here is the variation in flower and fruit morphology. And this is just a small subset of the total number of species, of 3 species that we can observe in this forest. And this is relevant because much of my work is based on the variation in form and function of this forest. And that was not that evident from the previous photo where everything was kind of green or unhomogeneous. But in general, these trees exhibit great variation, not only in flowers and fruits, but overall across all their different organs. So the general question that my research tries to address is, how do we get to these high levels of diversity? What are the main mechanisms that are maintaining this diversity? And I try to respond to this question from an ecological and functional perspective. So I try to understand how the differences in species strategies and how they use different resources ultimately shapes the way in which they are assembled in their communities. So for a long time, there is this recognition that there is not a single species that could simultaneously maximize all their functions and do well across all different environments, because there are constraints. And in the case of plants, we could think that the plants have limited resources for allocating and doing the different things that plants do. So we can think of that plants need to decide how to allocate, how to maximize their carbon fixation, transport water, or defend themselves against enemies, and many other functions. And because there are constraints, there are limitations in the resources that the plants have to allocate and distribute in all these different functions, is that we have the great diversity of species that we observe, right? So there is not one single optimal strategy that will do well across different environments, but instead we have different species that are doing well, that are doing different things, right? Because these constraints exist, and that overall generates diversity. So the idea of constraints was later formalized in this idea of trade-offs, and perhaps one of the best known and well-studied trade-offs is the one that describes the variation between R and K selected species, or the trade-off that describes the variation between fast and slow species. So based on this, right, we would have in one extreme species that are the slow species that have slow life history strategies, such as slow growth or small reproductive output. And then on the other extreme of this trade-off, we will have the opposite combination of traits, species that have fast growth and high reproductive outputs, right? This trade-off, the slow-fast continuum, has been central in many ecological studies. Here I'm just showing a few examples. For those, some of them all studies, and other ones more recent ones. But of course, tropical ecology did not escape to this idea of the slow-fast continuum. So here, what I'm showing is the existence of this slow-fast continuum, explaining the diversity of tropical forest in a system in Barro Colorado Island in Panama, in a tropical forest in Panama. So in this case, the trade-off is represented by the variation in tree growth and tree survival in this axis. And what we observe is that they form this negative relationship here, where each of these points represent a different tree species in the forest. So for tropical forest, this fast-slow continuum is associated with the way how plants are using the light in different ways. So typically, the species that are the fastest species that are in this extreme right here, that attain fast growth, are also the pioneer species, the opportunistic species that take advantage of the canopy gap. So when a canopy gap is formed in the forest, these species are the first ones to colonize, and they grow fast because they have all this access to light. But then in the other extreme, we have the slow species. These species tend to grow slower because they are limited by light. They are growing in these very dark, shaded understories of the tropical forest. And so we see that overall, right, we observe this variation between slow and fast species. And it seems that this trade-off seems to explain part of the diversity of the system. However, it seems that it is not enough for explaining this high overlap of species that we have in this extreme of the axis. These are the slow species in the forest, but also represent the vast majority of the species in this tropical forest. And it seems that this slow-fast continuum is not enough for explaining the diversity that is happening here, and we see a lot of overlap. So in my talk today, I'm going to tackle this problem from 3 different angles. First, I'm going to talk about the importance of a scale dependency in the constraints that form these type of trade-offs, and how will that help us to explain part of that diversity of the slow species. Then I'm going to talk about the redundancy in traits. That means multiple traits doing the same function, and that will also help us to explain part of the morphological and functional diversity that we observe in this forest. And last, I'm going to talk about the coordination across multiple functions. So in plant functional ecology, perhaps one of the best studied and most important trade-offs is the one that describes the variation between carbon conservation and carbon acquisition. This trade-off is analogous to a slow-fast continuum, and basically says that a species cannot simultaneously maximize the acquisition and the fixation of carbon with the conservation of carbon. So that could be explained by the variation in these 2 axes. So here we have the maximum photosynthetic rate of a species. So if species have high maximum photosynthetic rate, those will be the acquisitive species. They are efficient at fixing nitrogen. And then the other axis, the conservation axis, here is represented by the leaf lifespan, for how long those plants can retain those leaves, and that would represent the conservation of carbon. So what this trade-off and what theory says is that a species, again, species that maximize those 2 things are not possible, because there are some key evolutionary constraints, but then instead they should show this trade-off that I'm showing here. So in this extreme, we have the slow or conservative species, those ones that can retain those resources, that retain the leaves for a long time, but then the cost is that they cannot have an efficient photosynthetic rates. And then on the other extreme, we have the opposite set of traits. So what's the evidence that we have for these trade-offs operating in plant systems? What I'm showing here is a paper that was published in the early 2000s, a global study that included species from all major biomes, including tundra, tropical forest, deserts. And what these figures are showing is that there are these consistent and tight trait-trait relationships that overall support the existence of this carbon conservation and acquisition trade-off. So what we see here is that all these different leaf traits, leaf nitrogen content, leaf mass per area, leaf lifespan, right? All of these traits are ultimately related to these 2 axes that I just presented in the figure before. So because I know that these ideas are a little bit abstract, the constraints, trade-offs, and traits are a little bit abstract, I like to visualize all these through a synthesizer panel. So maybe this helps to understand a little bit better all these different ideas that I'm putting together. So the synthesizer panel, right, produce different sounds, and those will be the different functions that we observe in these organisms. And then we have all these different knobs that we can move and produce some variation in those sounds, right, in that function. So in our case, those knobs will represent the different traits that we observe in these plants. But then in addition to that, I'm adding these rubber bands, and those represent the constraints. So basically, if we have one knob, and we try to move one of these knobs here that is tied in this, through this rubber band, what that would generate is the movement in another knob that is also linked to that rubber band. So overall, that will make this a strong and consistent trait-trait relationship that I was showing in the figure before. So basically, those figures, that is why is that those trait-trait relationships, right, ultimately reflect the existence of the trade-off. But then one of the characteristics from this global study is that the trait values represented in these figures were traits at the species level. So each species was only represented by one single trait value. And this makes sense because this was the idea with this study was to evaluate the existence of these trade-offs at global scales. But when we are trying to understand what are the mechanisms that are assembling communities and what are the trade-offs that are operating in communities, maybe it's important to take into account that within the species variation that we know exists. Because ultimately, the composition of these communities is the outcome of interactions that occur among individuals, and individuals are never identical in their phenotype, are never identical in the traits. And here is just an example of how variable traits within a species could be. So thinking about the idea of constraints across different levels of variations across and within a species, what it is often assumed is that those trade-offs operate consistently across different levels of organization, and that is what I'm showing in this figure right here. So let's suppose that we have trait 1 and trait 2 that show a positive correlation across different species. Here, different species are shown by different colors because there are some trade-offs that result in this positive relationship here. But then the assumption is that those same trade-offs should operate within a species, and that is exactly what we are showing here with those positive relationships also operating within each of those species, right? But what I'm going to propose here is that maybe those mechanisms shaping these trait-trait relationships not necessarily are the same across different levels. So we could have, for example, this other case right here. So across species, we still observe that positive relationship between those two traits. But when we take a look at what happens within a species, those trait relationships tend to disappear. And the idea behind that, right, is that maybe the mechanisms that are forcing the trait-trait relationships in this case do not operate with the same strength or are not the same within a species. So for example, across a species, right, across different environments, the environment itself is forcing these two traits to work together, but there is not any additional physical constraint that will make these two traits to work together within a species, especially if those species are not distributed across wide environmental gradients. So to test these ideas, I collected intra and interspecific trait information from a tropical forest in Puerto Rico across 3 species that were widely distributed and common in this forest. So the species here were distributed along an elevational gradient from 250 meters and 1,000 meters. And what I did is that I collected individuals for these species, for these 3 species across different elevational belts. And for all of those individuals, I measured a bunch of different leaf traits, including leaf economic traits, the traits that represent this conservation and acquisition trade-off, but also other traits related to structure and to water use, all of them leaf traits. So in some cases, what I found, and it is not the result that I'm showing here, I found some consistency in these trait-trait relationships at both levels. That's not what was the most interesting result. So what I'm showing here are the cases, some of the cases in which those trait-trait relationships were not consistency across and within a species. So the black line in this case represent the relationships across species, and the different color lines represent the relationship within the different tree species, and these are the codes for all those different species that I measured. So what we observe here, let's say this plot right here, right, that is showing the relationship between leaf nitrogen content in the leaves and a specific leaf area. These two traits showed strong relationships at global scales. Those two traits are part of this trade-off between carbon conservation and acquisition, but I'm not always finding those type of relationships within a species. And what I think is happening here is that although these two traits are kind of working on the same type of functions for these plants, there are not any chemical or physical laws that are linking these two traits. So this is a chemical trait that represents just the amount of nitrogen that is in the leaf, while this one is a morphological trait that represents the size and the thickness of the leaf. And there is not any physical constraint, any physical law that will limit the variation of these two things. So perhaps across the species, across environment, right, there is a strong selection for these two traits to work together, but that is not necessarily happening within a species. So that additional variation, right, gives a little bit more flexibility for individuals that are interacting at the community level. So to summarize this part, I'll say that constraints shaping trait variation can depend on a scale. So assuming a single universal trade-off may overlook important biological flexibility. So if we go back and think out those slow species, that graph that I showed there, I was representing species as single values, and that graph didn't include any information about individuals, right, any within a species variation. What I think this work is contributing to is that it may add, the fact that there are some additional flexibility in those constraints may add some flexibility and may reduce part of the overlap, the great overlap that we are observing in those slow species. So that was the part, the first part about the scale dependency and constraints. Now I would like to talk about the redundancy in traits. But before diving into the science, I would like to acknowledge the many people behind this project. So this is a group of researchers, of Colombian researchers working in tropical dry forest, who were generous enough to allow me develop this project and supported the ideas that I'm going to present now. So thanks to all of them for their collaboration, which was essential for this project. So the project that I'm going to talk about now is based on a slightly different type of system. These are still tropical forests, highly diverse forests, but these are tropical dry forests that experience a strong seasonality in rainfall. So what you see here is exactly the same site, but in different seasons. This is the dry season, and this is the wet season. And because of the importance of water limitation in these systems, most of the work that I'm going to present now is based on water use strategies for plants. So we are going to switch from carbon to water. So the work here is based on a network of permanent plots that are that have been established in different patches of tropical dry forest. You can see the location of those forest plots here. So for all these different plots, we have information on species composition and also their dynamics, so their survival and growth over time. For a subset of those plots, we also have that are the points that are highlighted in red here. We also have information on the regeneration layer, so that's the seedling composition and dynamics. So again, we have information on the composition of all the species at the community level for those plots. And for all of them, we have been collecting trait data that represents the major resource uptake strategies for all these trees. But again, now I'm just going to talk out the traits related with water transport that are mainly represented by wood anatomical traits. So those are the ones that I'm going to talk about. So for plants, it has been proposed that one of the key trade-offs that describes the variation in how plants move water through their stem is described by the safety efficiency continuum, or the safety efficiency trade-off. Again, this is another analogous to the slow fast continuum. In one hand, we have a species that prioritize safety. That means that they prioritize or they reduce the risk of creating bubbles in their vessels. The vessels are the main cells that are moving water from the bottom to the top. And on the other extreme, the efficient species are the ones that prioritize efficient movement of water, but then that comes with the cost of having a higher risk of embolism or of a higher risk of creating bubbles in the stems. So the first step that we did for the project in this tropical dry forest was to evaluate whether this trade-off was actually operating in our communities. And this was a work that was led by my PhD student, Juan Celi. And what he did was that he measured the hydraulic conductivity. So this is a physiological measurement of how efficiently plants are moving water through their stem. And we related that to different vessel traits. So, well, and another thing that I want to mention is that many of the drawings that I'm presenting today are also from one of my students, Tomas, who is great at helping visualizing all these different functions that I'm talking about on plants. So, so Juan measured this physiological measurement that is basically telling us how efficiently plants are moving water, and then we try to evaluate what were the wood anatomical traits that were related to that. So this axis right here is actually a multivariate axis that represents the variation in two key vessel traits. One of those traits is vessel diameter. So if we move towards this side of the axis, vessel diameter will be increasing, and the other trait is vessel density that is negatively correlated to vessel diameter. So if we move towards the other side, vessel density will increase. So what we found is that species that have large vessel diameter and those vessels occur in lower density are the ones that have a more efficient transport, water transport in their stem, while plants that have small vessels have lower efficiency, but they have a safer movement of water, right? They have lower density and larger density and smaller vessels. So the idea here is just that small vessels, just by physics, have a lower chance of creating bubbles, but also having multiple vessels in the stems kind of creates parallel pathways for moving water. So if one of those gets an air bubble, gets embolized, we could get the replacement, right? So overall, that also would represent some safety. So here we have this combination of traits will be the safety, the efficient part of the axis, and this one here would represent the safety part of the axis. But then, when we took a look at what was happening with other vessel traits that we were expecting to be strongly related to this safety efficiency axis, we did not find the type of relationships that we were expecting. And that's the case of the pit diameter. So this was a project that was led by my postdoc Peter Williams. So pit diameter refers to the holes in the walls of the vessels that allowed the transport of water between vessel and vessel. So basically, within the stem, there are multiple vessels, and water flows through all those different vessels from the bottom to the tip, and are the pits the ones that allow the movement of water between vessel and vessel, as I am showing here. And there is a wide variation in those pit diameters, right? So what we were expecting is that, well, that pit diameter will control water flow, and smaller pits will represent much more like a safer strategy, right? Smaller pits will reduce the probability of creating those bubbles in the stem. So we were expecting that peat diameter will relate positively to vessel diameter, representing, so species with large peats and large vessels would be the efficient species, and then will relate negatively to vessel density. However, what we found is that that peat diameter was not related to either vessel diameter or vessel density. So what I think this is suggesting again is that through evolution, plants are experiencing these selecting forces for dealing with low water availability, right? And through evolution, there are multiple traits that have evolved to respond to that selective pressure. One of those is vessel diameter, but then another one is peat diameter. And again, there is nothing that is constraining the variation on these two traits. These two things are kind of going and responding to the same selective pressure, but there is nothing that is linking them physically. There are not any biophysical constraints that will make the variation in peat diameter and vessel diameter to be linked to each other. So what this is suggesting is that at the community level, we have some redundancy in traits. So in this case, small vessels and small peats will kind of result in equivalence in function, will result in safety in terms of how plants are moving water. And this will allow the formation or the existence of alternative solution, alternative phenotypes that are equivalent in their function. So in this case, hydraulic safety can arise from different trait combinations. We can have, in one case, one phenotype that has relatively large vessel size but small peats, and the functionality of this phenotype will be equivalent to this other one that has larger peats but a small vessel size. So going back again to this question about this low species, part of that great diversity that we observe in that extreme is the result of functional equivalence, right? There are multiple forms that we observe in this tropical forest that are working exactly with the same function. Now, moving beyond the vessel traits, there are other potential traits that might also contribute to expand the variation in how plants are moving water. Some of those traits are related to parenchyma. So most of what we know about plants, about how plants are moving water, is based on vessel traits, because those are the main cells that are moving water in plants. But in the stems, we also find other types of cells, such as the parenchyma cells that are mainly used for storage. And what we wanted to do in this study was to evaluate whether the parenchyma somehow would also contribute to try to explain the great diversity of form and function that we observe in this tropical dry forest. So this was a work that was led by my postdoc Andres Gonzalez, and the trait that we are studying here is called the vessel to parenchyma connectivity. So within the stems, we have the vessels that are here represented with this blue color, and then surrounding those vessels, sometimes we have some parenchyma cells that here is represented by those three different types of green color. So, and that connectivity between vessels and parenchyma is the trait that we are studying here, that is represented in this figure here. So there are two hypotheses that has been proposed to explain why is that there is a huge variation across the species in this vessel to parenchyma connectivity. One of the hypotheses proposes that this trait, vessel to parenchyma connectivity, is related to transport efficiency. So plants that have higher vessel to parenchyma connectivity will have higher or more efficient transport of water. And the idea is that the parenchyma that is in contact with some vessels, the parenchyma will release some solutes into the vessels that will change the osmotic potential and overall foster much more efficient movement of water. The other hypothesis is kind of going in the opposite way, more in the safety extreme, and suggests that this vessel to parenchyma connectivity will help to recover the functionality of some vessels that have some bubbles, some air bubbles. So the idea here is that the parenchyma will release some sugars into the vessels that have bubbles, and that would overall help to recover the functionality of those vessels. So based on these two different hypotheses, we have these different predictions. So for the transport efficiency, we were expecting that high vessel to parenchyma connectivity will result in an overall higher hydraulic conductivity and will be related with species with larger vessels as well. And that will also result in a faster growth. So species that have more efficient movement of water overall will have higher metabolism that will ultimately result in faster growth. For the hydraulic recovery part, we predicted that high vessel to parenchyma connectivity will result in higher survival. So the possibility to have some vessels recovered in function would prolong the life of these plants. So that is how we were expecting higher survival. And here are some of the results. So we found that vessel to parenchyma connectivity was not related to either vessel diameter or hydraulic conductivity or growth that I'm not showing here. The different colors here represent the species coming from different plots. But yeah, the main idea is that the relationship was flat. So what we think this suggests is that there is not a strong support for the hypothesis that vessel to parenchyma connectivity is related to transport efficiency. However, what we are finding is that vessel to parenchyma connectivity was positively related to survival and also was much more common in plants coming from the drier tropical dry forest. So the combination of these two results tends to suggest that perhaps that vessel to parenchyma connectivity in our sites is more related to hydraulic recovery. So this will be kind of an additional parallel way in which plants can get to that safety transport, but instead of using any of the vessel traits that I mentioned before, now the parenchyma will be contributing to that function. And in addition to the individual traits that I have been talking about, we are also finding that there are additional functional dimensions, that there are additional trade-offs that might be also contributing to expand the space for how plants are using water. And that's the case of the trade-off between fiber fraction and parenchyma fraction that was orthogonal to the safety and efficiency trade-off. So in addition to parenchyma and vessels, plants also have in their stems fibers. And the main function of these fibers is to provide mechanical support to the plant. So what we are finding is that plants cannot maximize the allocation of these two things. They could either maximize the allocation for higher fiber fraction or to higher parenchyma fraction. And what we think is happening here is that this higher allocation to parenchyma fraction, which, as I mentioned before, parenchyma is used for storage, might also contribute to help or to expand the range of strategies for using water or for dealing with low water limitations. So plants with higher parenchyma fraction can either store more water, which is important for when water is limiting, or store non-structural carbohydrates that are also used in the cases when plants need to shut down when water limitation is high. Plants shut down, stop doing photosynthesis, and sometimes they start starving because they don't have more carbon, right? So they could use those storage of non-structural carbohydrates present in the parenchyma fraction. So basically, what I'm suggesting here is that in addition to the trade-off that I just mentioned before, the safety and efficiency trade-off, here there is another independent dimension, right? That is explaining how plants deal with water, but now in terms of whether they are able or not to store more water or non-structural carbohydrates for dealing with low water availability. And what we are finding is that that axis is actually promoting survival across our different plots. So plants that have higher storage capacity, higher fractions of parenchyma fraction, they tend to have higher survival. So again, this would suggest that plants that have the ability to store water during these limiting conditions of water availability, then that they will have a higher chances of survival. So to summarize this part out, parenchyma, we found that there are additional key traits influencing water transport. That's the case of the vessel to parenchyma connectivity linked to hydraulic recovery, and that will represent kind of an alternative way for getting that functional equivalence. And that beyond the safety efficiency trade-offs, there are other hydraulic axes, and that's the case of the parenchyma and fiber fraction trade-off. And now to move into the last part, I wanted to talk about coordination in different functions. So for the first part of my talk, I was mainly talking about functions related to carbon. And for the second part, I was talking mostly about functions related to water transport. But actually, plants need to deal with these two things at the same time. And there has been some frameworks that have been proposed that suggest that plants should be coordinated in how they deal with these two limitations. So based on this framework, there is this idea that there should be a worldwide fast-slow economic spectrum where efficient hydraulic strategies should be linked to more acquisitive carbon strategies, and those would represent the fast species. And the safer species, in terms of their movement of water, will be also the more conservative in terms of the carbon strategies. And the mechanism explaining this slow-fast continuum here will be the photosynthesis, right? For fixing more carbon, water is needed. So species that are fixing carbon more efficiently will need more water for that, and that would explain this slow-fast continuum here. However, for the communities in the tropical dry forest, we are not finding that strong coordination between those two functions. So this is a multidimensional, a multivariate analysis that shows how all those two sets of traits are varying. So these traits represented in blue here represent the hydraulic traits, and what we see is that they are mostly loaded in this x-axis, while the carbon traits that are represented here by these green boxes are mostly loaded in an independent orthogonal axis, the y-axis here. So this suggests that these two axes are not coordinated for these communities, that instead they are decoupled. And what is more important is that these different sets of traits have real ecological consequences in determining the composition of these communities through different demographic processes. So this is what I'm showing here. So here are the relationships between those two sets of traits and two demographic processes, recruitment and survival. The traits represented in yellow represent the hydraulic traits, and the traits represented in blue are the carbon traits. So what we are finding is that most of those hydraulic traits tend to be significantly related to recruitment. So the species that tend to be safer in their water transport tend to attain higher recruitment in these communities, while for carbon traits, they tend to be mostly related to survival, but not to recruitment. So species with more conservative traits are the ones that attain higher survival. So what this result suggests is that there is not a single and unified strategy that is being optimized in this forest, but that instead these plants are kind of balancing these two different challenges. And these traits have independent impacts to different demographic processes that are different at different moments in the life of these trees. So to summarize this part, although photosynthesis depends on water, the traits controlling carbon gain and water transport can be decoupled at smaller scale, at the community level. That is the scale that I'm studying, and that different ecological filters shape them. Water traits are key for establishment, while carbon traits influence long-term survival. So again, if we go back to the problem of the slow species, what this part of my work is suggesting is that even though plants can show some overlap in one dimension, in one trade-off, what we are showing here is that, yes, plants are multidimensional. So they can still be different in other and independent dimensions, and that will foster coexistence. So if we go back to the main question that I introduced at the beginning of my talk today about how is that we can explain this high morphological and taxonomic diversity of tropical forests, I would like to kind of summarize what I think are 3 key points of my talk. First, that constraints vary across scales, within and across species, making well-known global trade-offs more flexible and allowing individuals to differentiate even when species converge on similar slow strategies. That multiple traits can fulfill the same function, being redundant, allowing the existence of alternative phenotypes with the same function, and that different functions can act independently, allowing species to differ in how they succeed, even when they appear to be similar in one dimension. So before I finish my talk, I wanted to mention that I did not talk about one important portion of my research, which is based on a slightly different type of system, is a forest that is under acute and chronic disturbance. I did not have time to talk about the projects on this system, but you probably will have the chance or already had the chance to hear out the work that we have been doing in this forest through the students in my group who had been working there. So actually a couple of days ago, Manar defended her master thesis and she worked in this system. She was interested in addressing questions about the role of disturbance on soil microbes and nutrient cycling. Tomás has been working on trying to understand the role of compound stresses, in particular droughts, severe droughts associated with El Niño and hurricanes, and how that impacts functionality in this forest. And Larissa is now thinking about integrating seedlings and adults in a unified framework for understanding the recovery process after hurricanes. So most of what we know about this forest comes from adult trees, but actually seedlings are a very important component of this forest and are also the future of the forest. And that is what we are thinking about for this particular project here. So you will probably hear more about it in the future. And to finish, I would like to acknowledge the many people that has shaped my academic journey so far. So first, I would like to thank to my advisor for their guidance, their patience, and for shaping the way how I approach to these questions on tropical forests. Also, I am grateful to the many friends and mentors and colleagues from EEB who have contributed to the work that I presented today in so many ways. They have enriched all these ideas in many different ways. Thanks so much for your support. I also want to thank my collaborators in Colombia who made possible the work that I presented today, and also are a constant source of inspiration through their hard work despite all the limitations in funding and many other barriers that are present in a country as Colombia. So thanks so much to all of them. Also, I would like to thank to the chairs who were very supportive from the very first day, and also to all the staff in EEB who had made possible all the work that I presented today and who have been kind of moving the threads behind the scenes. Thanks so much for all your support and work. And last, to my students, to the seedlings in my lab, thanks so much. To all of you, thank you for allowing me to be your mentor, and I have grown so much thanks to all of you, not only the ones that are present today, but also the PRAS members as well. So, and also to my funding sources who provided the funding to these projects that I presented today, and thanks so much for your attention. Thanks.
Apparently, it is our rule that on these promotion, celebration, seminar talks, we don't ask questions, which I think is a really stupid rule to try to keep scientists from talking science with other scientists. But you know, even though Nate's not here, if I allowed questions, he'd pop a vein or something. So that concludes the seminar, and thank you all for coming, and thank you, Natalia, for a great presentation.
Thank you. Thank you all.